So, in continuation with the previous lecture, now we have to see how proteins are modified in the lumen of the endoplasmic reticulum. As proteins are glycosylated on specific arginine residue at its end link, at its end terminal. The oligosaccharides consists of 14 carbon residues are attached to the accepted arginine residue of growing polypeptide chain as they are translocated into the endoplasmic reticulum. As they are translocated into the endoplasmic reticulum, the oligosaccharide is synthesized on the lipids here on the lipids dialcohol carrier anchored in the endoplasmic reticulum are membrane bound enzyme membrane bound enzyme called transferase. Oligosaccharide transferase four sugar residues here, four sugar glucose residues are added, four sugar residues are removed, and along with the one mano residue are removed, while still the protein are in the lumen of the endoplasmic reticulum. And the protein are still in the endoplasmic reticulum. So after getting modified in the lumen of the endoplasmic reticulum, then these proteins are sent to the Golgi bodies so that these proteins will be transferred to their destiny. So the export of proteins from endoplasmic reticulum to the ER, the whole pathway is called as the Secretory Pathway. Proteins travel from, as you can see also here, the proteins travel from endoplasmic reticulum to Golgi bodies in a vesicles. You can see these vesicles, small vesicles here. They form proteins travel in a vesicular form which forms the Secretory Pathway. These vesicles first bud off from one organelle, then move in a vesicular form and joins with the membrane of another organelle. Like here we can see the vesicles are coming from endoplasmic reticulum. Then they are they get first attached with the intermediate compartment. Then from intermediate compartment they will join the Golgi bodies. The intermediate compartment is the endoplasmic reticulum, which is not having ribosomes on its surface. It means this is the compartments which are present in the Golgi bodies and endoplasmic reticulum. This is the middle part where we are having smooth membranes on which ribosomes are not present. Many steps are involved in Secretory pathway, which involves vesicular transport between the compartments, which involves vesicular transport between different compartments. And then from Golgi bodies, as you can see here, the vesicles are either moving to join with the late endosomes. Then from late endosomes, they are able to form the lysosomes. Then another pathway these proteins are in, then they bud off at the trans side of Golgi bodies. They bud off in a vesicular form. Then they get attached with or they become the part of the plasma membrane here. Or another is they are having secretory vesicles. Then they fuse with the plasma membrane and the secretory proteins are thrown out of the cell. So there are some proteins which has to be there in the lumen. So we can say that some proteins which has to be retained in the lumen of the endoplasmic reticulum rather than proceed sorry rather than proceeding further rather than proceeding further. These proteins are like BIP, which are the chaperones, or the proteins which are the permanent resident of the endoplasmic reticulum are: first is the BIP, second is signal peptidase or the protein disulfide isomerase, and many other proteins are there which remain the permanent resident of the endoplasmic reticulum. The remaining protein which has to get exported to the Golgi body or retention in the endoplasmic reticulum makes the first branch point where there is sorting, so that proteins have to be in their correct destination. Similar branch point is present at each stage of transport, such as if we will talk about the protein which has to be which has to make the permanent resident of the Golgi body's lumen will stay there, or there some proteins from there will move and will be exported to the lysosomes or to the plasma membrane. For each point, we are having specific localizing sequences which are attached to these target proteins, which are present at to r- pr- to transport them their specific destination. To decide the destiny of the proteins, there are two types of target sequence. There are two types of target sequence. First is destined to the Golgi body or retained in the endoplasmic reticulum. The sequence for the endoplasmic reticulum, they are having KDEL. K stands for lysine, D stands for aspartic acid, E stands for glutamic acid, and L is the leucine. If this sequence KDEL is present at the carboxyl terminal, then the proteins will be in the lumen of the endoplasmic reticulum. The retention of some transmembrane protein in the endoplasmic reticulum is similarly short dictated by the short C-terminal sequence that contain two lysine residues. So we can say there is another sequence also which are having two lysine residue. Lysine is represented by K, so we will mention the double K, and some other amino acid will be present. So any other amino acid which will be present with the lysine will be represented by XX. So the sequence, if we are having KDEL sequence or KK and XX sequence, then these proteins are meant for the lumen of the endoplasmic reticulum. KDEL and KKXX signals do not prevent soluble endoplasmic reticulum proteins from being packed into the vesicles and carried to the Golgi bodies. So as you can see here, there are some proteins with the blue color diagram with the blue color ink, that these proteins are moving if by chance in case of bulk transport they are coming in the vesicles, then they fuse with the Golgi body, cis side of the Golgi body, then from the cis side only they are again sent back to the endoplasmic reticulum by the recycling pathway. Proteins bearing the KDEL and XX sequences appear to bind to the specific recycling receptor in the membranes of these compartments and are then sent back from Golgi bodies to the endoplasmic reticulum. This action indicates that This action indicates that there is a non-selective bulk flow of proteins through the secretory pathway, which leads from the endoplasmic reticulum to the cell surface. So, via this bulk pathway, the proteins are continuously coming from lumen of the endoplasmic reticulum. Then they become the part of the Golgi bodies, and from the Golgi bodies, protein are sent back. So, this was about the transportation, the secretory pathway. The third, last important point in this is the smooth endoplasmic reticulum and its role in the lipid synthesis. The smooth endoplasmic reticulum and its role in the lipid synthesis. On smooth endoplasmic reticulum, lipids are synthesized in eukaryotic cell. Due to their due to extremely hydrophobic nature of these lipids, these are synthesized with existing cellular membrane rather than in the aqueous membrane of the cytosol, because these lipids are hydrophobic in nature, so they are not synthesized in the cytosol. Eukaryotic membrane is composed of lipids, phospholipids, glycolipids, and cholesterol. Most of phospholipids are derived from glycerol. Most of the phospholipids are derived from the glycerol. These are synthesized on the cytosolic side of the endoplasmic reticulum from water soluble cytosolic precursors, where they form phosphatidylcholine, phosphatidylserine, and phosphatidylethanolamine or phosphatidylinositol. The fatty acids are first transferred from coenzyme A, carrier of glycerol 3 phosphate, by a membrane bound enzyme. Again, I will repeat, the fatty acids are first transferred from coenzyme A, carrier, to the glycerol 3 phosphate by a membrane bound enzyme. And the resulting phospholipid, phosphatidic acid, is inserted in the membrane. The enzymes of the cytosolic phase of the endoplasmic reticulum then catalyze the addition of different polar groups, which results in the formation of the lipids. The endoplasmic reticulum also serves as a major site for the synthesis of two other membrane lipids. As we know that our lipid also contain cholesterol and ceramide. The ceramide is converted to either glycolipids or sphingomyelin. Only membrane phospholipids are not derived from glycerol in the Golgi apparatus. Endoplasmic reticulum is also responsible for the synthesis of final or precursor of major lipids of eukaryotic membrane. So this was the about the role of the smooth endoplasmic reticulum in the lipid synthesis.